Uh, Question number one. Smilex China is a herbaceous plant. Figure 1.1 shows a transverse section of a root of S. China with root hair cells visible. Okay, sure. Name the type of microscope that has been used to obtain the image in figure 1.1. So if you can see closely, we are able to see the we can only see the outer cell walls or basically the cells at whole. We can see the minute contents of the cell, right? So that means that it's a light microscope. And if you like, you've done the practical, P3 or any practical in college, uh, yeah, bro, you are allowed to talk if you have doubts. No issue. We'll go chill, bro. So if you ever use a microscope, so that's how the transfer sections of usually transfers look like, right? So we can safely say that light microscope has been used. Like if this is almost into 84, right? So into 100, that's microscope. Calculate the actual length in millimeters of the root hair cell label in figure 1.1. Use the image length of the root hair cell along the XY. Okay, so we know that the magnification is equal to the image over actual, right? So that means the actual, the image over magnification. So right now, since it's not to scale like in the paper which we will have physically, so we can just estimate it. Let's say um, that this is around with the ruler over here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay. So according to the MSH I just checked, it's around 39 millimeter. So the image is if the image is 39 millimeter and the actual and the magnification is what 84, so divide 39 by 84 and you get um 1464 millimeter. Now and they've asked us to give in micrometer. So convert millimeter into micrometer, we can just like multiply it by we just open the millimeter, which is into 10 power negative 3, and then we divide by the unit which you want to convert in, which is 10 power negative 6. So basically, this 3 parts, and this since it's negative and the denominator, so it will go to the numerator to be multiplied. So we will just multiply by 1000, so we get 64 micrometer. Okay. Root hairs are important adaptations of root hairs are an important adaptation of root hair cells for the uptake of water. Explain one way in which root hairs adapt root hair cells for the uptake of water. So root hair cells basically they increase the surface area of the cell, right, to which is in contact with the soil. So more water can enter the cell through osmosis. So basically, yeah, they increase the surface area. This is a simple O-level question, nothing too technical, and it's 1 mark, so it's easy for absorption. Other points will include, um, let's write if the other points, so we can write if they reduce the diffusion distance. Let's say if it was either, this is 1 mark, right? So let's say they ask you a 3 mark question. So another thing you could write is uh, reach help in reaching a large volume of soil. So more volume of water is uptaken. Another thing which is like uh, very good if you write it is that is they increase the number of aquaporins. Aquaporins are basically special channels in the cell membrane which are specifically for the entry of water. Since um, since water is a polar molecule, right? H2O, it's polar, so it cannot directly diffuse through the cell membrane. So, and root hair cells need a large volume of water. So, what there is cells, there are aquaporins, so which facilitate the intake of water. So, if you like, this is a root hair cell, right? Let's say this is a normal cell, and this is a root hair. Since it's a root hair, it has a bigger membrane, right? So, since it's a bigger membrane, and since it's a bigger membrane, so it will have more aquaporins. Wait, what? Mineral ions are taken up by root hair cells. Table 1.1 shows the concentration of sodium ions and potassium ions inside the root hair cells of a plant root in the soil solution surrounding the plant, surrounding the root. Sorry. The reference to table 1.1 suggests the mechanisms involved in transfer of the sodium and potassium ions from the soil solution into the root hair cells. State the reasons for your suggestions. Just the mechanisms involved in the transport from the soil. So the only mechanisms which we have studied and which exist, no, bro, this is not leak. We don't do that stuff around that. So the mechanisms which we have studied are like diffusion, facilitated diffusion, and active transport, right, and osmosis. So let's first study the concentrations. So in the root hair cell, the concentration of sodium is 0.35, and in the soil it's 3.34. And for potassium, it's the opposite. So in the root hair cell, it is more concentration, and in soil it's less. So the mechanism for sodium is going to be facilitated diffusion. If you write simple diffusion, it will be wrong since we know that the phospholipid bilayer or the cell surface membrane, it's made of non-polar lipid molecules, right? Phospholipids. So they don't allow the diffusion of charged particles or ions. So what you need are transport proteins, channel proteins for facilitated diffusion. And so the reason, wait, how much see, right? Since concentration of soil is greater than concentration of root itself. Write this in simple words, I'm just like paraphrasing. And for potassium, it's the opposite. It's going to be, since in the root cell there is more concentration, so it's going to involve active transport. Concentration of cell is greater than. And if you like, you have none.
यार वो अब्दुल्ला, इसके अंदर हम ये नहीं लिखेंगे कि वो डिफ्यूजन नहीं, फैसिलिटेटेड डिफ्यूजन है, इसका रीजन भी लिखेंगे साथ कि फैसिलिटेटेड डिफ्यूजन क्यों है, क्या वो इम्पोर्टेंट
हाँ,
है?
बिल्कुल लिख सकते हैं।
वो आई थिंक आखिरी आखिरी में लिखना होगा, उन्होंने बोला स्टेट द रीजन। पहले आपको वो स्टेप्स बताना है, फिर आपको आखिरी में रीजन देना है।
इतना मैटर नहीं करता, आप आखिर में दो पहले दो, बट
हम
हाँ
लोग को
ये
पर
इसका।
वो भी मेंशन करना होगा ना कि कौन से प्रोटीन मेम्ब्रेन इन्वॉल्व है, जैसे फैसिलिटेटेड डिफरेंशियल पे आइदर बोथ कैरियर हो सकते हैं और चैनल, और एक्टिव ट्रांसपोर्ट में बस कैरियर
नहीं, वो
प्रोटीन।
आप वो करने की जरूरत नहीं है ना, क्योंकि देखो आप ये भी देखें ना कि आप क्वेश्चन वर्थ इट है या नहीं इतना चीजें लिखने का। आप अगर इससे जाए तो आप सारा सारा लिख दोगे यार, ये कौन साइंस करते हैं ये नहीं करते, बट इतना हमें जरूरत ही नहीं। हम देखो उन्होंने 3 मार्क है, तो 1 मार्क आपको मिल गया इसका फैसिलिटेटेड और एक्टिव ट्रांसपोर्ट का। दूसरा आपको मिल गया, ये आपने इसका बता दिया इससे कॉन्सेंट्रेशन बाय जाता है, और तीसरा आपको इसका मिल गया। 3 मार्क पूरे हो गए। अब यहाँ पे एक एक्स्ट्रा ये लिख दो कि आयंस कैन नॉट क्रॉस फॉस्फोलिपिड, तो आपको तो यहाँ
और
पे
ड्यू टू द
एक एक्स्ट्रा मार्क भी
हाइड्रोफोबिक
मिल
बोर्ड,
गया।
राइट।
हाँ, सिंस वो नॉन पोलर होता है और हाइड्रोफोबिक भी होता है, और क्योंकि बेसिकली आयंस क्योंकि आपका जो वो अनचार्ज है ना, और आपके बायर पोलर मॉलिक्यूल्स हैं। पोलर क्योंकि पोलर नहीं क्रॉस करता, तो चार्ज की क्या जरूरत है क्रॉस करने की, तो आयंस पोलर में ज़्यादा पसंद करते हैं ना। तो लाइक कोई कोई ऐसा वो स्टिमुलेंट नहीं होता उनको क्रॉस करने का, डिफ्यूजन में भी ना। कोई जरूरत ही नहीं है अंदर क्रॉस जाने की, क्योंकि बायर पोलर वाटर बायर एक्सटर्नल एनवायरनमेंट है वो आपका पोलर है, तो आयंस वहाँ पे फर्क करेंगे। The region between the outer layer of the root and the endodermis is known as the cortex. Table 1.2 shows the water potential of two adjacent cells, A and B, the cortex of a root. With reference to table 1.2, explain the direction of water movement between cell A and cell B. Okay. वाटर पोटेंशियल इसमें जो आपका कॉन्सेप्ट है, बेसिकली जो वाटर पोटेंशियल होती है ना, इसमें वाटर पोटेंशियल ऑफ जो आपका H2 है वो जीरो होता है Kpa, या जो उसका सिंबल है इस तरह कुछ होता है। So if the water potential of water is zero, and we know that water, or like pure, let's say it's pure water, right? Pure water. So there is no higher water potential than of pure water, right? So all other water potentials of any kind will be lower than uh, the water potential of water. So that means that the lower the water potential, the more negative its magnitude. So if we analyze the table A over here, cortex cell A has negative 120 and cortex cell B has negative 350. So we can see that the 351 is smaller than negative 120, right? It's basically this scenario, right? So, and we know that water travels to osmosis from higher water potential to lower water potential. So, uh, we say that the potential movement is from A to B, since water potential A is greater than water potential of B, and spread by osmosis.
हाँ है, एक क्वेश्चन था। ये नहीं लिख सकते कि वाटर पोटेंशियल ऑफ कॉर्टेक्स सेल ए इज लेस नेगेटिव देन कॉर्टेक्स सेल बी, दैट्स व्हाई
हाँ,
मतलब कि
या
जो
एग्जैक्टली वो भी लिख
इवेंचुअली
सकते हो,
फॉर्म होता है।
बिल्कुल लिख सकते हो, 100%।
ओके।
Okay, Muzammil, let's first finish this question, then we will go to a hard question. Okay. Some types of soil are made of small negatively charged clay particles that attract and bind to positive ions, such as ions, Fe2 plus. Fe2 plus that is bound to clay particles cannot be absorbed by. Okay. This reduces the concentration of free F2 that is available in soil solution population. Some plants that grow in soils. Uh, Sorry guys, the timer has come up, so I'm just gonna end the meeting and then restart it as soon as I end it. So just rejoin it when I end the meeting, right? So that it doesn't give us a timer after it ends officially. This basically it's just a hack. Sorry, I'm gonna end it now. Just rejoin the meeting, right? Okay. Assalamualaikum.
Yeah.
शुरू करो ब्रो, शुरू करो।
Oh yeah, मैं तब से म्यूट था बोल रहा हूँ मैं तब से बोल रहा हूँ अच्छा sorry. So basically the question has told it itself that the carbon dioxide will be reducing the pH of the soil. Since it says explain how the carbon dioxide released by the respiration can increase the concentration. So we know from the table it shows us that the lower the pH of the soil, the more the concentration of free Fe2 plus ions in the soil. So uh, the only way carbon dioxide increases the concentration is by decreasing the pH, right? So we can say that the CO2 release decreases pH of the soil by, I think it uses the concept from the uh, transport in blood, right? When the uh, carbon dioxide since so okay. We can write by producing, sorry,
कार्बोनिक एसिड के बारे में बताया था।
हाँ कार्बोनिक एसिड, बट यहाँ पे हमें लिख सकते हैं, यहाँ पे पॉइंट मिल जाएंगे मार्क्स, बट हमें वो कार्बोनिक एसिड बनते हैं ना, अगर कार्बोनिक एनहाइड्रेस हो, हमें पता नहीं यहाँ पे है नहीं, बट अगर आप लिखोगे तो आपको इसका मार्क मिल जाएगा। By producing H plus ions, which causes soil causes clay particles, sorry, not soil. Particles to lose Fe2 plus ions, since H plus displaces Fe2 plus, since it's more concentration, right? Okay, but
Bro, how does carbon dioxide produce H plus ions?
वो ये हमें पता, बेसिकली पीएच जो आपका लोअर करता है, एसिड लोअर करते हैं ना, तो पीएच, जो पीएच होता है आपका, बेसिकली मेजर होता है आपकी कंसंट्रेशन ऑफ H प्लस आयंस की। तो द फैक्ट दैट कि आपका कार्बन डाइऑक्साइड इंक्रीज करे कंसंट्रेशन की, और वो हमें पता है कि वो सिर्फ तभी हो रहा है जब ये पीएच ऐसा कम हो रहा है। तो वो आपका एक लाइक पीएच तब कम ही तब भी कम होगा जब H प्लस आयंस आपके उसको सोल्यूशन को मिलेगा ना। जो हम लाइक इन केमिस्ट्री व्हेन यू से दैट दी when you what does acid do? Acid produces H plus ions in aqueous solution, and we know that the acid reduces the pH of solution, right? So that's उसकी वो फैक्टर है कि वो हो ही हुए तब हो सकता है जब H plus ions आप प्रोड्यूस होंगे, pH
यानी
कम ही।
बेसिकली हमें भी नहीं पता कि एच प्लस साइंस आ कहाँ से रहे हैं, बस हमने खुद से बना के लिख लिए।
नहीं, हमें ये पता आ रहे हैं। हाँ,
आ
वो नहीं
कैसे रहे
पता
हैं, ये नहीं पता।
एग्जैक्टली, वो नहीं पता वो वैसे हम, चलो हम ये लिख देते हैं, बस फॉर्म कितने हम लिख देते हैं, हम क्योंकि यूजुअली जो हमारे पास हमने पढ़ा ये है, हम उन्हें सजेस्ट का है ना, सजेस्ट एंड एक्सप्लेन। तो
ओ यार,
सजेशन में हम अपना अपने वो नॉलेज का हम इस्तेमाल कर सकते हैं कि CO2 reacts with H2O to form carbonic acid। जो मैं सजेस्ट कर सजेस्ट हमने बस अपने नॉलेज से बताना है, तो हमारी नॉलेज में का ट्रांसपोर्ट मैमल्स वेले में यही आता है ना, कार्बन डाइऑक्साइड जो बोर इफेक्ट है बेसिकली उसकी, उसका डिफरेंट वो क्वेश्चन दिया, एसिड विच डिसोसिएट्स टू फॉर्म H प्लस। जो कि आपके चाहो, dissolved Fe2 plus is transported across the tissues of the root to the xylem. When the amount of dissolved Fe2 plus absorbed by root hair cell is greater than the amount that is needed by the plant, not all the Fe2 plus that is absorbed is transported to the xylem. Suggests how the endodermis can reduce the amount of Fe2 plus reaching the xylem. Basically, वो आपका this is the concept of the Casparian strip, right? We have an endodermis. Uh, so over here, at the endodermis, there's a suberin, Casparian strip made of suberin, which basically stops the pathway of water from the cell walls, which is the apoplastic pathway, right? And it forces it to follow the symplast pathway or through the cytoplasm. So over there, when the when water is only passing through the cytoplasm, that means that ions will not pass through, or excess ions cannot pass through at least. So we write that the Fe2 plus must cross the cell surface membrane. Endodermis due to the Casparian strip, and because of that, with the Casparian strip, which forces water and Fe2 plus into symplastic root or the cytoplasmic root. So here Kathi marks. And if you additionally, what you could have written was that there are only limited number of Fe2 specific membrane proteins. so which also reduces the quantity of Fe2 plus passing through the Symplast pathway.
दूल्हा जो एपोप्लास्टिक पाथवे है वो सेल वॉल्स के थ्रू होता है ना?
जी, सेल वॉल से थ्रू एपोप्लास्टिक होता है और सिम्प्लास्ट होता है आपका सेल मेम्ब्रेन से। बेसिकली आयंस है ना, तो आयंस तो आपके क्रॉस नहीं कर सकते सेल सरफेस मेम्ब्रेन, उनको प्रोटीन मेम्ब्रेन प्रोटीन चाहिए होते हैं, तो वो आपकी इतनी नहीं होंगी, इस वजह से कम जाएगा आपका। टू पार्ट ए, 2.1 is an incomplete diagram of the structure of an alpha glucose molecule. We figure 2.1 to show the structure of an alpha glucose. Okay, so we know that uh, the only difference between the alpha and the beta glucose molecule is this OH right here, right? In alpha, the OH is below the plane, and in beta, it's above the plane, and the rest all is same. So low, below, up, and below. So yeah, if they would have the beta glucose molecule, it looks something like this. Cellulose, starch, saccharide, sulfur glucose, right? Okay, and this is beta glucose. See, so in alpha glucose, the OH is below, and in beta glucose, OH is above the plane. Figure 2.2 shows part of a glycoprotein molecule. Say the types of, okay, glycoprotein, okay. So the types of covalent bond labels C and D. So it's a glycoprotein, right? So glycoprotein is basically a protein which has a carbohydrate chain attached to it. So in proteins, we know that the bond between two amino acids is the peptide bond, which is C, and D is a glycosidic bond. Many glycoproteins in the cell surface membrane are involved in cell signaling. State the role of cell signaling. State the role in cell signaling of glycoprotein cell membrane. Basically, the glycoproteins act as cell receptors, right? They are cell surface receptors. That's the main role which they play. One of the proteins found in milk is B-casein. Molecular B-casein consists of a single polypeptide, approximately 200 amino acids. So it only has tertiary structure, no quaternary structure. Molecules of B-casein have a high proportion of the amino acids proline and leucine. These amino acids have hydrophobic R regions. Okay, so that means that it is a globular protein since it has hydrophobic R region. Figure 2.3 compares the structure of proline with the general structure of an amino acid. This shows that proline is an unusual amino acid because it has a cyclic R group. Yeah, that's what I just observed. It's pretty unusual. And the nitrogen atom is attached to only one hydrogen. So it's a very weird amino acid. When a molecule of proline becomes part of a polypeptide, the hydrogen atom is lost from the nitrogen atom and is therefore not available for the formation of hydrogen bonds. Molecules of B-casein have a relatively low proportion of the amino acids cysteine and serine. Figure 2.1 shows the R groups of cysteine and serine. अच्छा, तीन को उन्होंने दे दिए मांग है। लो क्वेश्चन, लो ना खतरनाक क्वेश्चन। Just let me check what's the issue. Yeah, it's not loaded. Let me find the question real quick. Basically, exactly no question. That's the question. Compared to other protein molecules with a similar number of amino acids, weak acid molecules have a much less organized secondary structure, relatively little tertiary structure. The reference to the four named amino acids, choline, leucine, and cysteine, suggests possible explanations for these two observations. Okay, so it says that it has a less organized secondary structure, and we know the secondary structure is basically the primary structure is basically the sequence of amino acids, which is due to the peptide bonds, and secondary structure is the two forms, which is the alpha helix or beta pleated. So, and that is formed mainly due to the hydrogen bonds. So, it has a much less organized secondary structure, since it told us in the question itself that uh, when a molecule, the hydrogen atom is lost in hydrogen, and so it doesn't form any hydrogen bonds. So, let's organize. So, since proline didn't give us leucine, right? Proline cannot form hydrogen bonds with the CO double bond, since it is the main primary uh, component of the polar group. So this is positive, which no. Okay, that's how it forms hydrogen bonds. Uh, for the amino acids, and since there is a high portion of protein, so there is less hydrogen bonding in the polypeptide, hence there is a less organized secondary structure. High bonding and less alpha helix or beta pleated sheet formation. It has a relatively little tertiary structure.
यार अच्छा है ना, पर तुम तो क्या आउटलो
Okay. So we know that the uh, pro you know that the R group. Okay. Yeah. So they have a relatively low proportion of the amount of cysteine and serine. And since um since there is a relatively low proportion of cysteine, right? So that means there will be very few disulfide bonds and low proportion of serine. That means that the uh, yeah. So serine is also involved in hydrogen bonding, right? But since there is high proportion of you can get recording this, I'll send it in the groups where I sent it in inshallah soon. So basically, there is little cysteine. So there is less disulfide bonding for the tertiary structure. And we also know that the similar R group is not charged. Um, don't think that's the valid reason. They told us that it's a hydrophobic R group, right? So that means that it is non-polar. But if it was charged, so that really uh ionic bonds. I think that should be a valid point though. I think, but I don't see it in the MS though. But it should be a valid point. I'm saying trying to see where it said it cannot find form hydrophobic R group. Yeah, these amino acids have hydrophobic R groups. Yep. So since proline and leucine have hydrophobic R groups, so yeah, we can write it. You're right. So that's right. Proline and leucine do not form ionic bonds. So that's tertiary structure. Yep, it's a valid point. And since it's and the only thing it tertiary structure is dependent on is hydrophobic R groups. Right. Hydrophobic interaction. This is an extra point. I think that was sufficient. Okay, question number 3. Here 3.1 and 3.2 are diagrams of transverse section of the human heart during different stages of the cardiac cycle. The sections pass through the heart at a level that is just above the valves. So we can see the two atrioventricular valves. This is the bicuspid, so it is the left heart, and this is tricuspid. So this is the right side of the heart, and these look like semilunar valves. Name the valve label V. Basically, we just uh, term that these are semilunar valves, right? Since both of them are the atrioventricular valves. With reference to the chambers of the heart and the main blood vessels, describe the flow of blood to the heart that occurs when the transverse section of the heart appears as shown. So transverse section in figure 3.1, right? In 3.1, we can see that the semilunar valves are closed and the transverse no, transverse section basically means seeing the heart or seeing any sample by cutting through it, right? So transverse section of a stem is basically if you cut the stem either from the middle or from anywhere, and then you look it from that from the circular cross section, uh, like the circular part, you look at that from that angle, and basically there's a transverse section. So if you cut it vertically, like divided by two, uh, like let's say this is a stem, right? So um,
हॉरिजॉन्टली नहीं होगा.
क्या चीज
Transverse is basically cutting it horizontally, and longitudinal is longitudinal section will be cutting it vertically.
हाँ हाँ एग्जैक्टली, वो डिपेंड करता है ना आप किस तरह देखो, मतलब यहाँ पे अगर हम इसको इस तरह कट करें या अगर आपकी स्टेम है, तो जो आपका ये दो पीसेस आपके आगे ना, this you just divide by stem. So if you look at this this part, this circular uh, part from the microscope, that's what the transverse section is. And if you cut the stem longitudinally, so it means that if you're starting the whole of the stem, so basically what you get is you see the stem from the inner side as a whole. So you can see the contents of this, like all this xylem vessel and stuff, right? That's what you can see from the longitudinal. And in transverse, you can see the uh, top sections of the cell. So yeah, you can say it's top section if you mean it that way. With reference to the chambers, okay. So we know that the describe the flow of blood to the heart. So the blood, it flows from the when the some atrioventricular valves are open and the semilunar valves are closed, it is basically diastolic, right? So it flows from the atria to the ventricles, uh, or from and into the atria from the uh, different valves, which is the vena cava and the
क्या
pulmonary vein.
अब्दुल्ला ये पे एट्रियल सिस्टोल नहीं होगा?
हाँ, एट्रियल सिस्टोल नहीं, अब डायस्टली होगा ना, डायस्टली में भी तो आपके खुलते हैं, आप कह सकते हो। अ, दोनों में खुले होते हैं, एट्रियल सिस्टोली में भी, और अ, जो आपका डायस्टली होता है ना, उसमें उसके अंदर भी थोड़ा सा ट्रिकल डाउन करता है आपका ब्लड ना वेंट्रिकल्स में।
ठीक है
हाँ,
ठीक.
एग्जैक्टली ब्रो, अच्छी अच्छी बेटी जाकर यार। So basically, this is the transverse section, and this is the longitudinal way to like uh, see. So basically, they use the humanized roots stem of a plant. So hopefully, you understand what the difference between transverse and longitudinal is, right? Circle diamond. So we can write the flow is basically from the atria to the ventricles. Oh no, this dude, man. Just let me finish this question. And we can also set it into the atria. The vena cava and pulmonary artery. And that's what coming from the lungs to the heart, right? Wait, the what? No, pulmonary vein. What am I getting? It's coming from the heart. Okay, so I'm just gonna end the meeting again and restart it so that we are safe from the timer at the end.
Yes, and सेम लिंक होगा. लोग मेरे ख्याल से वापस नहीं आ रहे हैं, उनको लगेगा कि दूसरा लिंक आएगा.
Yeah, okay, ठीक है, same link होगा इंशाल्लाह. Just click the same link again. Inshallah. Assalamu alaikum. Identify the state of the cardiac cycle shown in figure 3.2. Figure 3.2, the atrioventricular valves are closed and the semilunar valves are open. So that means that the stage is ventricular systole. Since blood is being pushed into the aorta and the pulmonary artery. The reason is simple. Semilunar valves are open. Or you can write the atrioventricular valves are closed. Mark each.
और अगर सेमीलुनर वाल्व बंद होते फिर क्या होता?
फिर आपका डायस्टली होता है या एट्रियल सिस्टली होता है, जो यहाँ पे सीन है ना 3.1 में. मतलब ये कि अब आपका जो ब्लड ये है एओर्टा में वापस नहीं आएगा. आपका हार्ट रेट रीलोड कर रहा है, रिफिल हो रहा है ब्लड से. पहले एट्रियल फुल होएगा, डायस्टली के अंदर एट्रियल फुल होता है, फिर एट्रियल सिस्टली स्टार्ट होता है. जो एट्रियल में ब्लड आया होता है उसको वो सारा वेंट्रिकुलर में फेंक देता है, जिसमें ये आपके वाल्व्स खुल जाते हैं एट्रियोवेंट्रिकुलर. और फिर जब वेंट्रिकल फुल होते हैं तो वो एट्रियल वेंट्रिकुलर सिस्टम स्टार्ट करते हैं, जिसमें आपके ये वाल्व्स बंद हो जाते हैं एट्रियोवेंट्रिकुलर और आपके सेमिलुनर खुल जाते हैं, ताकि ब्लड आपका बॉडी और लंग्स में चला जाए.
हाँ, समझ आई थोड़ी सी अच्छी सी.
आगे
यस.
चलो. अच्छा, अब ये है और इशू इशू पे इशू लोड क्यों नहीं हो रहा है. व्हाट?
क्या है मैं ब्रो, पेपर ही फारिग था ये.
तो यही होता है. आई थिंक ये ट्रैक थैंक्स. हाँ ठीक है. चलो कोई नहीं, हम यहाँ से क्वेश्चन चुक लेते हैं. व्हाट्सएप से. 1 second. The rate and rhythm of the heartbeat are controlled by an area of specialized muscle tissue in the wall of the right atrium called the sinoatrial node. Describe the sequence of events that control contraction of the ventricles during the cardiac cycle. Basically, this is simple like the cycle of a heart, how the it contracts, why it contracts at specific time, and for that you need to know that the what the sinoatrial node is basically it's a myogenic muscle, right? So it produces electrical impulses which lead the atrioventricular node, which is in the septum, and over there is a short delay, and after that short delay, around 1.0.1 second, the AVN sends impulses to the Purkinje tissue, Purkinje fiber, and the impulses then travel down the septum, reaching the base, and then from there they travel upwards to the walls of the ventricle, and which causes the ventricles to contract at the same time. That's what you basically need to write. So we write the uh, since it's asked us to control the contraction of ventricles, so we write the impulse from SAN. Electrical impulse. So that's what it actually is. Reach the atrioventricular node after a short delay. Second. You need to know that the whole cycle of a heart, one single heartbeat takes 0.8 seconds. That's why the normal heartbeat of a person is 72 beats per minute. Since if you like divide 1 minute by 0.8, 60 by 0.8, you get 72, right? So in 160 seconds, it will beat 72 times. Impulse sent to Purkinje tissue. Purkinje tissue is basically uh, this here right here. This is the AVN. This is the SAM. This is Purkinje tissue. And from the Purkinje tissue travels to then travels to base the heart, and from there the impulse travels upwards, causing ventricles to contract. From base upwards, this is very important because the uh, seminal valves of the aorta is basically positioned upwards, right, not downwards. Both ventricles contract at the same time. I just wrote all the points which are possible, and you only if you write only three of from these, so you'll get full marks as well. बेसिकली यही होता है मैंने भी आपको दिखाया यहाँ पे ऐसे एन है यहाँ पे बनता है आपका सिग्नल ट्रैवल करता है एवीएन एवीएन से बंडल ऑफ हिस वहाँ से परकाइन टिश्यू इसकी ज़रूरत नहीं है ये पता होने की बस आपको परकाइन टिश्यू एवीएन और इसका बताना चाहिए वहाँ से बेस पे बेस से फिर ऊपर और आपका यहाँ से शुरू होता है कॉन्ट्रैक्शन और यहाँ तक एंड खत्म होता है तो हमने ब्लड को यहाँ ऊपर भेजना है hence that's the reason. Well, I lose marks if I didn't write in first round upwards. If you wrote, no, you won't lose marks. Like if you wrote that the impulse reach AVN and then the Purkinje issue, and then you write the Ponticus contract at the same time, so you will get full marks, hopefully 3 marks. Just need to get 3 points, right? So you have a margin. But But to be on the safe side, just try travels upwards because just travel the all sequence, right? The all events occurring in sequence. So hopefully don't lose any marks just to be safe. Let's just go to the next question. नान ये आपका 2025 है February March 2022. Question number 4. Vaccination programs are widely used to help control the spread of infectious diseases. State-wise, some diseases are described as infectious. They are described as infectious since they can be transmitted. Not all diseases are transmissible. So infectious since they are caused by a pathogen or transmissible. पेपर का थ्रेशोल्ड क्या होगा? पेपर का थ्रेशोल्ड भैया से अल्लाह जानते हैं कैम्ब्रिज वाले बाकी पता नहीं यार, it's not very difficult compared to other past papers I've done. I think around somewhere in 40s, like
No, around 35 होगा इसका क्योंकि काफी इजी है ये वाला पेपर।
हाँ तो इजी है तो थ्रेशोल्ड ऊपर होगा ना।
ओ हाँ 40 42
तो
मे बी।
हाँ 40 ये लो 40 ना, इनिशियल 40 होते हैं, पता नहीं I don't
लाइक
know.
मैंने इससे मुश्किल पेपर्स देखे हुए हैं, उनका 40 था। ये तो बहुत आसान लाइक ये वाला।
उस साल लाइक ज्यादा बच्चे होंगे अब हम
हाँ
जैसे कम में आ गए तो इतना नहीं होगा इंशाल्लाह। ना खैर करें। Person was given an injection to give protection against infectious disease E. Person has not previously been infected with disease E26 is the second injection to give protection against disease was given. Concentration in the blood of the antibody specific to disease was measured over a period of 60 days from the time of the first injection. Show the concentration of the antibody in the blood over the period of 60 days. Okay. Basically, think the second injection is a booster shot. So concentration of the antibody. 1 second. I'm sorry, it's just we have to face these problems when you don't have the PDF. But I'm gonna. The concentration of the antibody in the blood after the second injection was higher than the concentration of the first injection. Explain why the concentration of the antibody in the blood was higher after the second injection of the antibody concentration high इसलिए होगी क्योंकि आपके उन्होंने फर्स्ट इंजेक्शन में लाइक सिर्फ प्राइमरी रिस्पॉन्स हुआ है ना, लाइक लेट्स सपोज इंजेक्शन में आपका वीकेंड पैथोजन था, तो फर्स्ट इंजेक्शन इज द प्राइमरी रिस्पॉन्स, then so then after the first injection सेकेंड इंजेक्शन में अब आपका सेकेंडरी रिस्पॉन्स प्रोड्यूस हो गया। पहले आपका प्राइमरी रिस्पॉन्स में आपके अच्छा कुछ बी लिम्फोसाइट्स प्लाज्मा सेल्स बनाए, मेमोरी सेल्स बनाए काफ़ी ज़्यादा ना, सो देन द सेकेंडरी रिस्पॉन्स अब आपके जो पहले मेमोरी सेल्स जो इंक्रीज हुए थे आपका इंक्रीज चांस भी इंक्रीज हो गया, लिम्फोसाइट्स भी इंक्रीज हो गए, तो अब आपके प्लाज्मा सेल्स इंक्रीज होंगे एंड देन दे प्रोड्यूस मोर antibodies. So that's what it is. So we say that the memory cells were produced after the primary response, which was the first infection. So more lymphocytes.
यानी वो दूसरे जब बूस्टर होता है उसके अंदर यानी वो होता है बेसिकली नॉट वीकेंड प्रॉपर होता होगा पैथोजन ताकि पूरा इम्यून रिस्पॉन्स आ सके।
नहीं, पूरा पैथोजन तो फिर वो इन्फेक्शन ही है ना वो। मतलब आपको वो क्यों वायरस देंगे? मतलब कभी ऐसे नहीं करते कि आपको असली वायरस आपको दे दें, क्योंकि फिर तो आप बीमार हो जाओगे। वो तो हमने वही से तो हम बचना चाह रहे हैं। तो वीकेंड पैथोजन यही होता है, पैथोजन होता है। तो हमें हमें पैथोजन तो नहीं चाहिए होता ना, पैथोजन नहीं हमें चाहिए, हमें पैथोजन के एंटीजन बेसिकली चाहिए होता है। तो बेसिकली वो आपको एंटीजन प्रोवाइड करता है, और लेकिन आपको वो इल नहीं करता। तो
हाँ
वैक्सीन
ठीक है।
भी होता है। वरना पैथोजन भी अगर लाइव पैथोजन दे दें तो फिर आप ऐसे ही ले लो। किसी जो खांसर हो तो उसके साथ बैठ जाओ। इंजेक्शन के पैसे क्यों देने? वो लिम्फोसाइट्स,
तो हम
रिसेप्टर्स।
टाइम कैसे मैनेज करें बायो के पेपर में? एक बायो का पेपर कंपैरेटिवली ज्यादा लेंथी होता है केमिस्ट्री और फिजिक्स से।
यार वैसे अगर आप देखो ना तो टाइम हो जाता है। आपने बस ये करना है कि ना, यूजुअली होता है ना जो क्वेश्चन, मतलब अच्छा आप देखो, चाहे क्वेश्चन वन कर लिया आसान है, अब टू पे आएगा। टू अजीब सा क्वेश्चन दिया, अब आप सोचने लगे अच्छा क्या करना है। अगर देखो, अगर 5-10 सेकंड दो, मैक्सिमम 15 सेकंड, नहीं उस वक्त आंसर आ रहा ना तो टाइम ना जाया करो, बस आगे चले जाओ, छोड़ दो उसको। हर पेपर का यही सीन होता है, छोड़ दो और बाकी क्वेश्चंस करो। वहाँ पे क्वेश्चन 4 भी मुश्किल था, वो नहीं हुआ, छोड़ दो, 5 पे जाओ, फिर वापस क्वेश्चन 2 पे आओ, होपफुली हो जाएगा। नहीं होता छोड़ दो, फिर
बायो
से क्वेश्चन 4 पे जाओ।
बायो में तो क्वेश्चंस भी ज्यादा होते हैं बायो के अंदर।
हाँ वही ना। तो ऐसे तो होता नहीं कि सारा क्वेश्चन आता हो, वुमन वो एक पार्ट होता है जो तंग करता है, तो पार्ट स्किप कर दो और आगे करो। जितना पेपर मैक्सिमम एक गो में कर सकते हो करो। फिर वापस ना, जैसे सर साथ कहते हैं थे लाइक, आप बार बार चीज़ें रिपीट करो, बेहतर होता है कि आप एक दफ़ा फुल लगा के करो ना। आप बार बार पेपर को ना गो थ्रू करो, स्प्रिंट लगाओ ना, अच्छा ये हो गया, ये हो गया, ये हो गया, ये नहीं हो रहा, स्किप कर दो। और हमेशा ब्रो, मैं बता रहा हूँ, मेरा 100% रह के जो क्वेश्चन आपको उस वक्त नहीं आ रहा होता ना, और आप उससे दोबारा आते हो तो तब आपको आ जाता है वो। और तब भी नहीं आता उसको फिर छोड़ो, फिर आगे जाओ, फिर दोबारा आओ, और फिर तो 100% आ जाएगा इंशाल्लाह।
थैंक यू।
तो एंटीजन, सो मोर प्लाज्मा सेल्स for increased production of antibodies. And you can also write that the uh, increased chance of memory cell encountering pathogen antigen, since there are more memory cells in circulation from the primary immune response. Second, second person was given a different injection. Second person was given a different injection to give protection against another infectious disease F. Person had not previously been infected with disease F. With concentration in the blood of the antibody specific to disease F was measured over a period of 60 days from the time of the injection. Okay. So okay. State the type of immunity that results from the injection given as protection against disease F. Okay, so
artificial
कौन सी
artificial active
artificial active? You sure?
oh passive I guess
हाँ ये artificial passive क्योंकि basically यहाँ पे you know the injection at the time of injection there's a like surge in concentration of antibody and that means that just injected antibody in the body, right, in the person. You didn't give him any pathogen. From here we know that at the time of injection there is no antibody. That means you've given them the antigen of the pathogen so that they develop the antibodies themselves, which they do. But in this case, just gave them the antibodies. So it's basically artificial passive. The artificial passive, the other types are basically artificial active, right? That's just quick glance. Immunity. Yeah. So active immunity, when there's a production of memory cells, that's what active immunity basically is. So natural active is basically when you get infected, so that the body comes in contact with the pathogen and they develop memory cells themselves, or you give them artificial, which is by vaccination. And then you have passive immunity, which is pre-made antibodies or and which is temporary. Natural passive is from the breast milk, basically the initial substance in the breast milk, which is colostrum. It's a yellow type substance which is released in the initial days of breastfeeding, and that's what basically contains the uh, antibodies for the child. And then we have the artificial passive, which is pre-made antibodies, which you just inject them, which is made from the hybrid myeloma method, monoclonal antibodies. Like you provide antigen to organism, usually a mouse, and then you harvest the plasma cells and you fuse them with cancer, bring a hybridoma, and then you just culture the hybridoma to harvest the antibodies. So I haven't seen many questions for the hybridoma method anyways. The key features of the type of immunity resulting from the injection given as protection.
यार ये आर्टिफिशियल पैसिव क्यों होगा ये ऊपर हमने वैक्सीनेशन की बात नहीं की सारी हमने
एफ के पूछे ना, एफ में क्या हो रहा है, एफ में
इसमें
एंटीबॉडीज
ऊपर करना
हमसे
ओके ये दूसरा ग्राफ है ठीक है
हाँ ग्राफ लिख रहा है।
मैं उसके ऊपर वाला ग्राफ देख रहा था ठीक है
हाँ नहीं है होगा। तो आपकी डिसाइड फीचर्स ऑफ टाइप ऑफ इम्यूनिटी इंजेक्शन, फीचर्स इसके ये होंगे कि देयर इज अ टेम्पररी प्रोटेक्शन अगेंस्ट the vaccine, so it's a temporary protection. That means that the no memory cells are produced. And its effect is immediate. In 2021, the number of new cases of tuberculosis was estimated to be 10.6 million. Name the bacterium that caused tuberculosis. So we have two bacteriums. Both names start by Mycobacterium and Mycobacterium tuberculosis or bovis. Bovis is basically found in cattle. TB is often treated with several different drugs at the same time. This is necessary to kill multiple drug-resistant strains of bacteria, which is lengthy, taking 6 months or more to make sure all bacteria are killed. The care cause TB react to the presence of rifampicin. Rifampicin is an antibiotic often used in successful treatment of tuberculosis. अच्छा उन्होंने क्या किया
टाइमर कितना रहेगा?
क्वेश्चन जो है आपका वो है कि just and explain how knowledge gained from this research could improve TB treatment and reduce the chance of rifampicin resistant material. Basically यहाँ पे हमें ये समझ आ रही है कि जो आपका रिफैम्पिसिन होता है ना antibody often used as a treatment TB, right? So basically the bacteria in this culture क्योंकि वो आपका रिफैम्पिसिन को पंप आउट कर रहा है दैट मीन्स इट्स रेजिस्टेंट लाइक वो अपने आप को बचा रहा है रिफैम्पिसिन से उसके पास एबिलिटी है तो अगर हमने उसको उसके उस रेजिस्टेंस को हम टैकल कर सकते हैं वो भी उन्होंने बता दिया कि फर्दर रिसर्च और अब कॉमन ड्रग्स जो आपके इंडाइजेशन को ट्रीट करते हैं वो आपके बैक्टीरिया की रेजिस्टेंस को भी डिक्रीज कर रहे हैं मतलब वो अब उसको बाहर नहीं निकाल पाएंगे रिफैम्पिसिन को तो मतलब वो अपनी इफेक्टिवनेस इंक्रीज हो जाएगी बैक्टीरिया के अंदर रहेगा अंदर वो गड़बड़ कर देगा बैक्टीरिया की तो फिर हम ये लिख सकते हैं कि वी कंबाइन द ट्रीटमेंट राइट तो हाउ नॉलेज so we combine both drugs and they ask how so we say that the uh, with indigestion drugs to keep this antibiotic inside bacteria so that it kills bacteria more effectively. And then you can also write that the this will and then they have asked for treatment and if it kills the bacteria so that means that it will have less time for mutation to occur. Okay, so initially the bacteria aren't resistant, it's just a other property of the bacteria. Resistance is when like they won't be affected by rifampicin at any cost. resulting in resistance to the campus. अच्छा अमित ब्रो मैं बीटेक बंद करने लगा फिर से, तो ज्वाइन करो ठीक है।
तो ब्रो बात सुनो,
हाँ
लाइक मेरे ख्याल से इतना बहुत है।
पक्का।
हाँ, लाइक मैं बस थोड़ी सी अभी नोट्स एक बार रिवाइज करने लगा हूँ वही, और
हाँ वैसे सही कह रहे हो ब्रो, सही कह रहे हो।
हम्म।
बेस्ट ही है क्या रिमोट।
उसके बाद आपको कितना थैंक यू करूँ, काम है आपने टाइम निकाला अपना।
और नहीं ब्रो मैं तो वेरा इंसान हूँ एक समय मैं
नहीं
रिकॉर्डिंग तो
यार,
बंद करूँ ना ये, प्राइवेट।